In this video we will discuss about a lipid membrane exchange strategy to optimize the IL-2 based nanoparticles. So basically it is a well-known fact that in the in vivo system or within the mice or in the human beings, this lipid membrane exchange occurs because of the serum or albumin. So they want to use this lipid membrane exchange in the in vivo to optimize their layer by layer based nanoparticle systems. So idea is that they take a membrane that is coated with different layers that includes IL-2 and also uh, one positive and one negative layers of amino acids that we have discussed in first video of this paper. And when they store it in the DIH2O, then they are in the condensed form. However, polymer architecture and rearrangement occurs when they move them into the physiological solutions, exposing IL-2, and thereafter they will traffic. Trafficking occurs to the tumor nodules, and where they will release IL-2 at a slower rate. And this will not only improve the efficacies of their nanoparticles, as it will expose IL-2s into the tumor nodules. At the same time, the rate at which IL-2s are released is slower, so that it will increase the lifetime of these nanoparticles within the tumor nodules also. And so they check two different conditions. First, they check the covalently bound nanoparticles and saturated it with uh, lipid, and the other case is their previously gold standard nanoparticle, that is Mal-lbl based nanoparticles, and this they have coated with lipids, but it is not saturated, and they inject these nanoparticles into the HM1 tumor bearing mice, and after 24 hours they took the confocal microscopic images, and they are able to see that in the case of Mal-lbl, the nanoparticles are spread across the cell membrane and also in the extracellular matrix. Whereas in the case of saturated LBL, where IL-2 is covalently bound, they are able to see that most of these nanoparticles are within the cellular matrix, or pocketed within the cellular matrix. That can be seen here. Here they use WHEA to stain cellular matrix, and we can see that the particles can be found in the cellular matrix. Thereafter, they also checked the effect of this lipid membrane phenomena on the tumors, whether they are able to suppress the tumors or not. So for that, they took this HM tumor bearing mice, and day 0 they considered it as the day at which they inject tumors to the mice, and at day 7 they inject this lipid, this IL-2 based nanoparticles with saturated lipids and saturated lipids. They also use the control that does not have IL-2, and the result shows that the controls or unloaded LBL does not have any impact on the tumors as expected, and for all other cases they are able to see a drop in the tumor bioluminescence. However, the reoccurrence of the tumors occurs soon. Reoccurrence of the tumors occurs in all the cases, except there is a delay in reoccurrence for this Mal-lbl case, and because of this delay, it has increased the survival rate of those mice to around 31 days, median of 31 days, whereas for all other cases, survival rates are low because of the reoccurrence of the tumors at early days, or in the early days after the tumor treatment. That can be seen here. So this indicates that their Mal-lbl efficiencies can be improved through this lipid membrane exchange. However, the covalently bound IL-2 is not capable of improving it any further compared to the control where they use free IL-2. And one more thing they are able to find out is that the compositions of the membrane plays a role towards its uh, diffusions of these IL-2s or nanoparticles. As we can see that Mal-lbl can diffuse evenly across the cell membrane and in the cellular matrix, whereas the covalently bound IL-2 is not able to diffuse across the cell membrane. They are pocketed in the cellular matrix in a very condensed region. So that's all for this video. I hope you understood to some extent how they try to optimize further the IL-2 based nanoparticles. If you need to understand more about this topic in detail, you can go to the reference that has been provided in the description. That's all for this video. Thank you for your time.